Hi, everyone. So my name is Riley Ennis, co-founder and chief operating officer at Freenome. And I'm here today to talk to you about not only how you can use genomic information to improve your own health, but really encourage everyone here to think about what products can tie the complexity that is your genome to information that really improves overall human health. Whether you know anything about biology, or you're already in the biotech space, or you're just interested in how to make a difference in people's lives, I really want to encourage everyone here to start playing around with the data that's available and start thinking about how we can make the genome more actionable. So being in the biotech space for a couple of years, I've become a hypochondriac to a certain extent. And the moment that 23andMe came out, I ordered the 23andMe test. And 23andMe is taking information from your spit and looking for genomic information. This genomic information is associated with variants. These variants are different mutations associated with changes in your genome. Here, I am sharing with you my actual 23andMe report. And what's really interesting is the piece that I learned from my 23andMe information was that I have a deficiency for this specific protein. Why does that matter? Well, if you do a little literature searching, you dig around, you read probably 5 or 6 publications, you realize that this protein, the alpha-1 antitrypsin, is associated with liver disease. So now, on an annual basis, I look at my AAT levels in blood, and I look for changes associated with AAT, which would predispose me to a liver condition. This is actionable, right? Am I going to dig through hundreds or even thousands of different variants and say, what can I do and change about my lifestyle, or about what am I monitoring with my health to see what information I can use on a day-to-day basis? Now, when you think about this, there's another problem. AAT as a variant doesn't necessarily guarantee that you're going to have liver disease. There's a disconnect between the genomic information and the clinical phenotype, or the manifestation of disease. So the challenge here is that although you can tease out little bits of information and update and change your lifestyle, unfortunately there isn't a perfect relationship between genotype and phenotype. I'm sure we all hoped that after sequencing the human genome that this was the reality, but it's just not true. The genome looks more like this. This is a representation of the whole genome called a Circos plot, and you can see that not only are we talking about thousands and thousands of genes, but they're highly interconnected. So what this means is that by understanding relationships between genes, understanding how we can take this information and correlate the complexity to a physical phenotype that we can see, such as wellness, lifestyle, or even diseases such as cancer, we might have the chance of bridging this gap. The impossibility, though, is the genome is very large. So what we have to do is start thinking about what can we learn, or what can we understand about the genome to connect the dots between the complex information and then a visual or larger representation of a clinical phenotype. So what's exciting is that in the image recognition space, when you're looking at distinguishing different types of dogs, you actually take a pixel, red, green, blue, ultraviolet, and you start to understand features or shapes within the picture that allow, through training a machine learning model, to separate pictures from one dog from another type of dog. The same principles apply to the genome. Actually, you can take some of the same algorithms looking at T, C, G, and A, which are the bases in the whole genome, and you can think of the genome as just this giant string of T, C, Gs, and As. You can then train models to start looking for different data structures and representations of genomic information tied to a label like cancer or no cancer. It becomes very simple then, without understanding biology or necessarily understanding all the details behind what information is being gleaned from genomic information, to separate different labels. So as long as you know what the problem is, you have the metadata and the clinical labels associated with information, you can start separating and training models to classify, in this case, disease from non-disease, which is very exciting. And I think more importantly, some groups have started to apply similar principles of just transforming the genome into a data type that is consumable by a neural network. So Google has been very successful with variant calling, being able to call variants within genomic information. And what they've been able to do is to take this information, train neural networks to predict variants with high sensitivities and specificities. The challenge still, though, is how do you then take the information and say, given the complexity and the variance associated, how can I change my lifestyle or become more actionable? So that even though we're applying data science to some of these problems, there's still a gap between using this information on a day-to-day or an annual basis. So some questions that we might be interested, and one that I think a lot of people overlook, is how is your genome changing? A lot of us think the genome is fixed. We send in our spit sample, the genome is what it is. If you are sitting out in the sun for too long, if you drink, there's all these lifestyle implications that can impact your genome through time. And we should start to use these tools to understand how your genome is changing. Additionally, can you ask a question like, what diet works best for me? Can we abstract away the complexity of the genome and then start asking questions that are relevant to what I'm doing with my lifestyle choices? And finally, is my genome healthy? Which is one of the problems that at Freenome that we're working on today. So some background on the company, and to show you some data on what we're working on in applying some of these methods to disease detection. In cancer in particular, early detection is very important because if the disease is localized, you can improve and actually prevent metastasis of disease. So early detection becomes very important. So we started the company about 2 years ago, and we're backed by Andreessen Horowitz, Founders Fund, Polaris, and some other groups. And we're really focused on bridging the gap between early detection and early intervention. So we've done now in ovarian, breast, lung, prostate, colorectal, we have preliminary data using some of the transformations I talked about to start to classify different disease types. And we're working with several academic groups and regulatory bodies to move these tests kind of through from the pre-commercial stage all the way through development is where we are today. And what you are seeing here, and what I'm going to train all of you to do, is to detect cancer. What you're seeing here are two medians across different representations of the whole genome. And visually, you can see the differences between somebody who is healthy versus cancer. You don't need to know all the individual genes or what variants are present, but if you're able to classify a macro signature associated with a clinical label, you can detect cancer, even with your own eyes. More importantly, and this was a part of kind of our early development work, is we're looking at how you can take, even within a labeled group of somebody having cancer, can you resolve and perform transformations that separate early from late-stage disease? This ties back to actionability in the sense that early disease is traditionally more of surgery, and later disease is chemotherapy. So not only does early detection have incredible utility, but in fact, using machine learning and really thinking about the genome of what changes occur in disease states, we can see separation between the early and the late stage disease, which is pretty profound. So what you're visualizing here is based on the features that we're focused on from a genomics perspective of these DNA fragments that we see in blood when you take a blood draw. You can see that there are differences that are pretty distinct between the later stage and the early stage disease. And moving forward, all the examples that I've showed you of the problems and I think how we're able to look for not only disease but other changes from just a health perspective is how we start thinking longitudinally. And I mentioned that the genome does change, but even from a statistical perspective, how do you classify a trajectory across genomic information is a huge challenge and something that in at least the biology community has not been solved yet, and where I would really encourage everyone here to really put a lot of effort and to start thinking about. And to show you what this looks like, what I'm going to walk through is a case study of somebody developing prostate cancer. You're seeing their Circos plot, which is a visualization of the whole genome. So these are all the chromosomes. And you see these little peaks like buildings that are regions of genomic instability, meaning the genome is somewhat different and there's a deviation from normal. Unstable is bad, so if you see a lot of peaks and your Circos plot looks like the skyline of Manhattan, then you're probably not doing so well. If the peaks look more like somewhere in the Midwest, then you're probably doing a little bit better. Now, what's interesting is in this individual, we calculate a metric called their molecular age. This individual was a 21-year-old from a Freenome perspective, but they were actually 40. And what you're going to see in these samples over time from blood samples that were collected in a biobank, in fact, the information that you can see over the years is pretty different as somebody goes through the aging process. Also, you're going to see PSA, it's a test for prostate cancer detection, compared to Freenome's test. So over the years, you see increase in genomic instability. The peaks start to increase. This is normal, whether it's sunlight, smoking, whatever you're doing in your life, that can just have an impact, and that's completely normal. What's not normal is what happened in the 2011 sample, where there was a jump in genomic instability. You can see that the peaks go up exponentially, and in fact, there was a mutation present in the sample that was associated with cancer. Now watch what happens over the next couple of years as this person's developing later stage disease, going from localized to metastatic. What's scary to see is without early detection, we let genomes continue to mutate and change over time. But I think what's even more empowering when it comes to actionability is by tracking these changes, not only can you understand trajectory and you can understand how somebody can change through time, we can start to ask the question of what can I do about this? Before, we had to look at all these individual genes and tie it to information that we know based on a diagnosis or a clinical label. But there's no way to track changes over time to see if I try a new diet or if I lose weight, am I actually improving my genomic health? And this is the key in terms of the future of actionability, is taking the trajectory of this information to drive disease prevention altogether. So I hope everyone here today, and look forward to engaging with you after the talk, but really using this information, thinking not about what are the genes, but how can we abstract away the complexity to build products that can really advance human health. Thank you.